So this is the structure and function of the lac operon. So the promoter, the operator, and these genes are all part of the DNA. Now, what the promoter does is it allows the RNA polymerase to start coding for these proteins. The operator is where the repressor can bind to, and these three genes code for proteins that help break down lactose. So the first thing that happens is somewhere either before or after this gene sequence, a repressor will be coded for, and the repressor in this state will bind to the operator. In this state, it can bind to the operator because there is no lactose present. And this is important because the repressor restricts the RNA polymerase from coding for the proteins down the line. And it does not need to do this right now because there is no lactose present, and these proteins that the RNA polymerase would code for are proteins that can break down lactose, but since there's no lactose present, that is not needed to happen. Then, when the lactose is present, what happens is lactose will bind to the repressor, and the repressor changes shape due to this chemical bond, and then this allows the RNA polymerase to go all the way down the line and code for certain proteins. And as it does this, a messenger RNA will be formed, and this messenger RNA will go off to code for this protein. In this case, it'll be the LAC-Z protein. Now, what this LAC-Z protein does is it can break down the lactose present. So it'll begin breaking these lactose molecules down, and eventually it will break down the lactose inside of the repressor, and there will be no more lactose present in the system. And when there's no more lactose present, the repressor returns to its initial shape, and it can bind to the operator once again. And once again, it will restrict the RNA polymerase from going down the line and and creating more LAC-Z proteins. And this is important because, again, there's no more lactose. It's not necessary to code for more LAC-Z proteins. So what we see here is this DNA system controls itself for when to and when not to code for certain proteins to break down lactose.